very good morning to all present here. Thank you for joining us at this time and your interest in this breakfast at UMHealth. And we're going to have our first speaker, Dr. Tang. She's our clinical microbiologist. She has been with us in this department for more than 8 years. And today, her topic is going to be on Campylobacteriosis, concerns beyond just food poisoning. So we may start the talk. I will pass it over to Dr. Suni.
Thank you, Professor Chandra. I will share my screen. So good morning, everyone. I'm going to share some thought about Campylobacteriosis. So Campylobacteriosis is the infections caused by Campylobacter. The most common species causing clinical infections is Campylobacter jejuni. There are a few other species as well. So let me begin with sharing two interesting scenarios, interested in such a way that our lab has encountered some challenges to confirm and to growing the species from the blood culture. So this as a routine day when as a clinical microbiologist or lab personnel, we go to the lab to look at those positive blood culture and do some troubleshooting or review the results. But on this day, things happen not so straightforward. So we have two cases here. Case number one is from the blood culture anaerobic bottle, which flag positive at day 4 of incubations. So as a recap, when we receive the blood culture bottle, we will incubate in the automated incubation machines. So for positive, the machine will flag alarm and we will take out the blood culture and perform gram stains. We call it direct smear to give preliminary results. And then culture is a gold standard for confirmation of bacterial infections. So for this particular blood culture, although the machine flagged as a positive, but from the gram stain, there is no organism seen. Occasionally, this can happen, but not that common. So we do have some false positive, but because culture is still a gold standard, so we routinely culture on those lab media, blood agar plate, McConkey plate, chocolate agar plate. However, there is no growth on all for this blood culture. There is no growth at all. We consider some organism may be slow grower and we extended the incubation for another day, another 48 hours. Also, still no growth. So we don't simply call it as a false positive. Being a clinical microbiologist, we also look into the clinical correlations, and based on our experience of handle this kind of cases, we do suspect some fastidious organism. So we look into the patient's clinical history in the electronic folder. For this patient, he is a 68 years old man with a few underlying comorbidity like stroke, diabetes, or that COPD. So the reason for this admission is he had symptoms of acute gastroenteritis. They complained of 2 episodes of loose stool on the morning of admissions, associated with nausea and fever, and history of taking outside food. And he had an episode of syncope. Claim is due to the lethargy after all these GI symptoms. Otherwise, according to the documentations, the patient was stable and baseline blood culture are not remarkable. So concerning this gastrointestinal tract symptoms and our previous experience, we do suspect it could be a Campylobacter, which is a common foodborne pathogen. So Campylobacter is very difficult to grow or not growing on those routine blood agar plate, McConkey, or that. So it requires a special selective media. So although this one is a blood culture, is a sterile specimen, but this Campylobacter is a selective media designed to isolate the Campylobacter from the stool sample because the stool is full of a diverse normal flora. So this selective media has the antibiotics to suppress other microbiota and also provide nutritional support to this fastidious Campylobacter. In addition to not only fastidious nutritionally, it requires special media, it also requires special incubations atmosphere, microaerophilic incubations. It requires a lower oxygen level, like 5 to 10%, 42-degree temperature, and prolonged incubation, not just an overnight incubations. We can incubate up to 72 hours. So for this blood culture, after 48 hours incubation, yes, there is an isolate growing and identify with the multitoph as Campylobacter jejuni. Multitoph is a mass spectrometry, is a advanced technology for species identifications of microorganism. So there is a second case also. This time is from the aerobic bottle, which FRAC positive at day 3 of incubations. So same thing also, this gram stain, we found it very difficult to locate the organism. So it looked like this. So this can be normal for most of the blood culture. Some inexperienced person or who never see any microbiology slide might mistaken this pink color thing as a gram-negative bacteria, but actually it's not. It's the background matrix from the blood culture. The blood culture bottle has the broad culture media and also some substance like resin to bind to the antibiotic. So in case the patient had received prior antibiotics, there are material to bind with that antibiotic so that it won't interfere with the culture. So all this can give rise to this morphology. These are not the bacteria. It could be very normal. You can see it and in almost all of the blood culture bottle. But for this positive blood culture, which FRAC positive, we saw a few very faint staining, very thin and spiral-shaped bacteria. It looks similar like the background matrix. So very difficult to distinguish whether it's the bacteria or the background matrix. So strong suspicion is required. So we, based on our previous experience also, we think that it looks like the gram stain morphology for Campylobacter. It's very difficult to see from here, but I can show the typical picture. So this is the view under microscope. So classically, the gram stain morphology of Campylobacter look like that. Classically being described as a spiral shape, some say comma shape or slender. So we think what we saw in that real microscopy look like this. So again, we look into the history. So this one more complicated. The patient is a 37-year-old lady with multiple comorbidity. So this is what we get when we are looking at that blood culture. The patient later on had a prolonged admission, prolonged staying in the ward with more recent investigations, especially on the renal pathology and other. She got other concurrently positive culture, like a positive urine culture as well. But for the purpose of this sharing, I will be focused on the gastrointestinal tract symptoms. She also complained of fever with chills for 2 days and a documented temperature of 39 degrees, 2 days diarrhea with multiple episodes, up to 7 episodes on day 2, and noted blood streak stool on day 3, vomiting and intermittent abdominal pain. Husband also had a similar complaint. So we also, especially based on the gram stain morphology, we also suspect Campylobacter, and we do the same thing, put in the special media, special incubation, and yes, after 48 hours, we isolate the Campylobacter jejuni. So this one is the Campy alga, black color because it contains charcoal, which provide better nutritional support for the Campylobacter. And as expected, it cannot grow on just a basic routine culture media. So we have successfully overcome those challenges. So result of both cases will convey to the clinician in charge as soon as we confirm the species. As far as I understand from the documentation, patient 1 was likely just had a simple transient bacteremia and treated as outpatient. Patient 2 was more complicated due to other underlying comorbid and some new investigation results. She was under the treatment care of infectious disease team. So from the microbiology aspect, what we have learned from managing or troubleshooting these two challenges are the ambiguity of the gram stain morphology that could be confusing or nearly being oversight. Gram stain is a very basic lab technique, a very old method, but still very useful to provide prompt preliminary diagnosis. I think the appearance of the spiral shape or seagull shape cells are quite typical. As long as we see that, we can provide a preliminary diagnosis, say this is possible a Campylobacter. Not many other pathogens present with morphology like this, but technically from our previous experience also, it's very easy to get oversight because it's looks similar with those background matrix and very faint staining. So even for a laboratory personnel, high clinical suspicion is required. And another challenges are it is fastidious to grow in the culture. We require those special culture media and prolonged incubation periods. In the past, some laboratory also struggle to achieve accurate species identifications. But nowadays, in our center, with the presence of multi-TOF as an advanced technology, so identification is reliable and I think no longer an issue nowadays. But there are other limitations. One of them is the antimicrobial susceptibility testing. Again, due to the fastidious to grow in the culture, after we spend like 48 hours to grow the organism from the blood culture, again, we need to spend another 48 to 72 hours to do the antibiotic susceptibility testing, and also lack of standardization in the testing panel. Usually the lab will do selectively tested on a few antibiotic agents, which are the treatment options. So for the patient number 2 just now, due to the more complicated underlying comorbids, so when the clinician requested to add the additional antibiotic susceptibility, which was a few days later, the isolates is no longer available. So it happened sometimes some organisms are non-viable or the isolate is no longer available in the lab. So I will proceed with some discussions. The concern is not just because this is a blood culture or invasive infection or bacteremia, but there are other concerns. The main concern is Campylobacter is also reported to be increasingly drug resistant to their antibiotic options. So this is a bit older data from the CDC. So as in 1990s, CDC already provides warning on the increasing resistance. Here say decreased susceptibility to ciprofloxacin over the in the past 20 years from these presentations. So Campylobacter is a zoonotic. Most of the time you get it by a foodborne route with associated with poultry like chickens or egg and some other animals. So these are the data shown by CDC a few years ago, already provided warning for reduced susceptibility to ciprofloxacin, azithromycin. Another is not here is the erythromycin, which are among the first line options for Campylobacter. So I went through a few relatively newer references. So most are still highlighting increasing multi-drug resistance, particularly against ciprofloxacin and erythromycins. So World Health Organizations and CDC also concerned about the resistance in fluoroquinolones and macrolides. So the antimicrobial resistance are thought to be associated with the antibiotic overuse in livestock and also veterinary sectors. As we know, Campylobacter is foodborne and zoonotic origins from those farm animal. So those are working as a reservoir for the resistant genes. So this review study, it reviewed the published data over the previous 5 decades, involved more than 500 Campylobacter isolates. The two most common species infecting human are Campylobacter jejuni and coli. So they also analyzed the genome data available in public database. So it's proposed that among these two species, the antibiotic resistance involve both chromosomal and acquired. Chromosomal means intrinsic. When the bacteria start to replicate, the resistant genes are already there and carry forward. But acquired is mobile genetic elements like plasmid. This is the similar mechanism as those more common hospital-acquired infections like ESBL, CRE, or that. The small mobile genetic elements enable the resistance to spread rapidly, not only among the same species and also cross-species. So this study also proved that horizontal transfer of the resistant genes, horizontal means from one species to another, are possible. So these are the data show that against the major classes of antibiotics, Campylobacter also show multiple types of the resistance mechanism, like beta-lactamase enzymes, multiple efflux pump, alterations of the antibiotic target sites, all that, and common resistant genes. Some of them also seen in other gram-negative bacteria. So this plasmid is the one that has the potential to cause a rapid spread of the resistance. So this is another recent study analyzed over 500 isolate of Campylobacter jejuni, performed the susceptibility testing using standard MIC method, and also perform whole genome sequencing and bioinformatic analysis to characterize the resistant mechanism and then correlate the bioinformatic analysis with the in vitro susceptibility testing. So this also correlate with the first review study just now, identify increasing percentage of resistance among the fluoroquinolones and macrolides, and also a few common resistant genes. And this one also support that or propose that in some other gram-negative, for example, bacteria or gram-negative bacteria, they may require the whole segment of the genes to encode for the resistance mechanism. But there are evidence that not only this article, but there are others who support that in Campylobacter, even presence of a point mutations may encode for the significance antibiotic resistance. So these work on fluoroquinolone, macrolides, and some aminoglycosides. So this is another references. This one is a comprehensive one, not only looking into the clinical isolates or human isolates, but also looking at the animal isolates and also Campylobacter isolate from the environment and across a larger geographical area across 53 countries. So this one analyzed or review more than 66,000 Campylobacter strains across the different continents and involve human, animals, and environment isolates. For the recent years isolate with genomic data available, they also perform bioinformatic analysis and they try to correlate the finding with factors like their socioeconomic status, climate changes, and the use of antibiotics are the contributing factor for the increasing or emergence of AMR. And because it's a large-scale study, so the result varies among different regions. For example, in some country, consumptions of certain antibiotic shown to have positive correlation with the AMR in human or animal isolate, but some did not show significant positive correlations. However, it also in agreement with most other reports, like common emergence of resistance in increasing resistance in Cipro and macrolides, and found a similar resistant mechanisms. So this is the fourth review. This one actually is a literature or report from the animal sector, from the veterinary. So this one, again, they also look at resistance against major antibiotic classes, and they found that the similar resistance, the clinical isolate and also isolate from animal share the similar resistant profile, and also some of the resistant genes are present simultaneously in clinical isolates and animal isolate. So this, again, the animal isolates can work as a, because the Campylobacter has a wide range of the animal reservoir, so it can be the origin, or it can be the increasing concern of the resistance. So the summary, increasing antimicrobial resistance among Campylobacter species is common, and of increasing global concerns is associated with the overuse of antibiotics, not only in the healthcare sector, but also the animal industry and vet sectors. Those resistant isolate in animal provide a wide range of the reservoirs. So two clinical important species infecting human are Campylobacter jejuni and Campylobacter coli. But there are a few rare species, but occasionally will cause the infections. So comprehensive surveillance and evidence-based control policy are required for a more comprehensive study on the resistant profile. And there are still a few challenges, including the wide range of animal reservoirs in the industry sectors or animal sectors, and the clinical lab isolations and confirmation of the pathogens are difficult, especially in resource-limited laboratory. Like just now, we need to use a special media, set up the special incubations atmosphere, and use a multi-TOF for accurate species identification. This may not be easily achievable in some resource-limited setting, and also with the concern of the AMR, antimicrobial susceptibility is very important, but again, it's limited with the subject to the limitations in the organism isolations, species confirmation, and also lack of standardizations in the antimicrobial susceptibility testing method. I think I finished my sharing sessions. Thank you very much.
Yeah, thank you, Dr. Suni. Thank you so much for your comprehensive talk on sharing the cases and also giving us the awareness on antibiotic resistance. We have two questions from Dr. Tan Cheng Yin. So the question is, so is blood culture better than stool culture in isolating the Campylobacter? That's the first question. The second question is, in cases of GBS, in which the Campylobacter infection is an antecedent infection, in that case, what would be the best way to isolate the organism?
Right. Thank you for the questions. For the first questions, whether blood culture is better than stool culture, in my opinion, actually, it depends on the clinical presentations. And actually, I think the challenges or difficulty is not about the specimen type, it's about the fastidious nature of the organism. It required the special culture media and also special incubation atmosphere. So the Campi alga, that selective media that we use, is designed with components and antibiotics to suppress the gastrointestinal tract flora. So I think it's still able to recover, give a good recovery from the stool. In our practice also, majority of our Campylobacter isolates are being cultured from the stool. The blood culture is when like there are invasive infections or bacteremia. I will say both are important, depend on the clinical presentations. So the second one, if your GBS means Guillain-Barré syndrome, because Guillain-Barré syndrome is actually immunology reactions, like the antibody against Campylobacter lipopolysaccharide also attack the subbrain, the tissue in the central nervous system. So actually, I think it's due to the immunology response.
Okay, thank you, Dr. Suni. So we have one more question. Is the specific Campy gel medium long-lasting, or does the bacteria also mutate to avoid detection? And may I know how often Campylobacter infection can be life-threatening?
About the sorry, can
Gel
you
medium.
repeat the first part of the questions again
Yeah,
about the
Campy gel
media?
medium long-lasting, or the bacteria also mutate to avoid detection. So
Oh,
basically, yes, the sensitivity
I
of the
see.
gel medium, Campy gel
Mm.
medium.
I think, according to what I understand and our practice, I think it's quite good to recover the Campylobacter jejuni, which is the main species that infecting human, whether it's long lasting, about the whether the bacteria mutate to avoid being identified. I think the mutation that we discussed just now is more to the resistance. The difficulty to be isolated or be culturable is again due to the fastidious, and also maybe if other stool normal flora overgrow. So I think we try to minimize it by using a few antibiotics in the selective media to suppress the stool microbiota. And
Okay.
whether how it causes severe infections, yeah, it can be seen in like immunocompromised or patient with low immunity.
And thank you for answering that. And I think the current, I mean, the last question that I'm going to ask is quite long,
Yes.
but I will, due to
Yeah.
time factor, I will just make it very short, and perhaps you can type later if
Yeah,
you have
sure.
more answer, I mean, elaborate answer. Okay, so you mentioned that fluoroquinolone resistance is common, but macrolide resistance can still happen too. From a micro perspective, do you have any local data on resistance trends?
For this morning presentation, I didn't prepare the data, but we do have
Trend
some
data.
data there. But I think it's also like what I mentioned just now, there are lack of standardizations in the antibiotic testing panel. So our testing is based on the first line options, or the uh, by request if the patient on certain antibiotic treatment. I think I see another
Okay.
question. Sorry,
I
I
think
think there's another questions. Oh,
yeah, but due
I
to
will.
time factor, is
Okay,
it okay
can.
we proceed and we can just type the answer instead? Is that okay?
Yes, sure.
Okay.
Okay.
Thank you for all the participants who have interest in this topic, and also asking so many questions, and really thanking you all for your virtual presence and participation. So with that, I will pass over to next moderator, Dr. Shaikh.